This is a GCSE Biology presentation on prokaryotic cells. To get started, I'd suggest you go and watch the presentation on eukaryotic cells if you haven't already done so. But if you are confident and you can label all the parts of a plant and animal cell and you can identify the function of each part in a plant and animal cell, grab yourself a scrap of paper, make a list of them now, pause the presentation, and when you're ready, you can go back over and check through your answers. Just be careful with the table on the screen. It's asking for plant and animal cell structures in one column and the structures that are only found in plant cells in the other column, as well as their functions. Pause the presentation now, and when you're ready, you can restart the presentation and check your answers. Pause now. Well, there are 5 main structures found in plants and animal cells that you need to be aware of. There's an additional 3 structures that are only found in plant cells. Again, if this is unfamiliar to you or you're not totally confident in this, I suggest you go back over the eukaryotic presentation before you go any further through this one. In this presentation, we're going to be looking at plants, animals, and bacteria cells and be able to identify which of those are eukaryotic as the plant and animal, and the prokaryotic cells are the bacteria cells, and how we tell the difference between diagrams of those. We'll also learn the different parts of a bacteria cell and making sure that we also are aware of the difference in eukaryotic, not just in terms of structure, but also in terms of size of the cells. So when it comes to the differences between prokaryotic and eukaryotic cells, the biggest difference is that prokaryotic cells have no nucleus. They have genetic material, they have single circular DNA molecules. These are on top of things we'll find out called plasmids as well, but they have their own circular DNA that is free floating in cytoplasm. They have no nucleus, whereas plant and animal cells, which are our eukaryotic cells, do have a nucleus. Prokaryotic cells and eukaryotic cells have ribosomes, but in prokaryotic cells the ribosomes are smaller. We do not refer to ribosomes as organelles. Organelles are not included in ribosomes. You can talk about mitochondria, you can talk about chloroplasts, but you do not talk about ribosomes when you're talking about organelles. Hence, there are no membrane-bound organelles found inside prokaryotic cells, but they do have ribosomes. Prokaryotic cells are much smaller than eukaryotic cells, and both prokaryotic bacteria cells and plant cells from the eukaryotes, remember, not animal cells, but plant cells from eukaryotic cells, both have a cell wall, but the cell wall in a plant cell is made from cellulose. In a prokaryotic cell, it is not made from cellulose. There are some extra bits in prokaryotic cells you need to be aware of as well. We have small circles of DNA called plasmids. These are as well as the other DNA that exists. There's also a layer called a capsule, and the capsule layer is found around the outside of the cell wall, and its purpose is to protect this prokaryotic cell, a bacteria cell, as well as to let the bacteria cells stick together into clumps. You'll also find that prokaryotic cells have tails called flagella, and most importantly, prokaryotic cells can divide by a process known as binary fission. Do not say that prokaryotic cells replicate by mitosis. They do not. There are no chromosomes involved. There's no nucleus involved. The prokaryotic cells divide by binary fission, and that's a very common exam question. What you're looking at now is a diagram showing the typical format for a prokaryotic bacteria cell. All you have to do is, using the information from the first bit of the presentation, identify structures 1 all the way up to 6. Pause the presentation, have a go at trying to label them, go back over the previous bit of the video if you need to, and when you're ready you can restart the video. Pause now. Well, the structures are as follows. The big thick layer around the outside of the cell is referred to as the cell wall. We then have the liquid inside, which is the cytoplasm, just like a plant and animal cells. We have no nucleus in this cell because it's prokaryotic, so we have got the genetic information. Not always DNA, so genetic information or genetic material is absolutely fine. Remember, we also have a cell membrane, and just like in a plant cell it's turgid, the cell membrane is pushed right up against the cell wall. Number 5, we have small circles of DNA known as plasmids. And number 6, we have flagellum. Now remember, flagellum are not always present. Sometimes a bacterial cell will not have those flagellum. What you can also have is you can also have a layer around the outside of the cell wall known as the capsule, which is not included in this presentation. So now that we've identified some of the structures found inside prokaryotic cells, you also need to know the function of each part. Pause the presentation, again go back and look at the diagram if you need to, but try and work out of all the different sections that are labelled on the prokaryotic cell, what is the function of each part. When you're ready, restart the presentation. Pause now. Well, in a bacteria, so now what you should be able to do is you should be able to recreate the table from the start of the presentation where you have the eukaryotic cells and the animal plant cell structures only, eukaryotic cells and the plant cells only structures, and then a third column for your prokaryotic cells, and be able to identify the structures in there as well as the functions of each structure. Pause the presentation, have a go at that now, and then restart when you're ready. Well, if you have been able to memorize it, you should have included the cytoplasm, cell membrane, cell wall, and the ribosomes, all found inside or around bacterial cells. The functions of each of those is exactly the same as the functions inside animal and plant cells. So you can use exactly the same wording for both. There are of course some extra bits in the prokaryotic cell that are not found in any of the eukaryotic cells. You have the plasmids, which are rings of the DNA which are linked to antibiotic resistance. You have the flagellum, which is involved in movement. And of course you have the capsule, which provides protection for the cell and also allows the cells to stick together with other cells. One of the things you're going to be expected to do on the GCSE exam is to be able to convert between different prefixes. Now, prefixes are words that we put at the start of measurements in order to change the scale of that measurement. So for example, meters is a unit of measurement, but we can have centimeters, millimeters, micrometers, and nanometers. When we have centimeters, 1 centimeter is worth 10 millimeters. 1 millimeter is worth 1,000 micrometers. And 1 micrometer is worth 1,000 nanometers. And you're going to be expected in the exam to be able to move from one of those prefixes into another. In order to do that, my suggestion is this. Start off by taking one of whatever the bigger unit is. In this example, we're going to take 1 millimeter. And then write on the other side of an equal sign how many of the smaller unit, in this case micrometers, we would get for that 1 millimeter. If we want to get from the number 1 to the number 1,000, we simply have to times by 1,000, because 1 times 1,000 equals 1,000. If, however, we want to get from the number 1,000 back to the number 1, we have to divide. And I would encourage you to scribble in the margins of your exam this little kind of diagram whenever you're trying to convert between units. Because if you want to convert millimeters into micrometers, you simply times your number by 1,000. If, however, you want to go from micrometers back to millimeters, you divide by 1,000. You will need to know those 4 names and how you convert from one to the other. If you have to go all the way from centi all the way down to nano, simply do it in 3 stages. So to finish off, what you need to do is create a set of questions or key that will allow you to help identify, when presented with a cell in the exam, is that an animal cell, a plant cell, which are 2 types of eukaryotic cells, or if the cell that you're looking at is a bacterial cell, which is a prokaryotic cell. So you're looking for a set of questions or some sort of key that allow you to identify which of those 3, A, B, or C, would correctly label your cell. Pause the presentation now and I'll show you an example when you restart the presentation. Pause now. So here's an example. Does the cell have a cell wall? Well, if there's no cell wall, we know immediately that it is most likely that that cell is eukaryotic and an animal cell. Now, there is an important way to check. If it is eukaryotic, remember all eukaryotic cells have a nucleus. If it does have a cell wall, then we know it cannot be a eukaryotic animal cell. That leaves us two possible options. It's either a eukaryotic plant cell or it's a prokaryotic bacteria cell if there is a cell wall. Well, the easiest way to spot which one's which is I can check for the nucleus, or I could even go and check for plasmids. If there is a nucleus, it is a eukaryotic plant cell. If there is no nucleus, it's a prokaryotic bacteria cell. If we're looking for plasmids, remember the plasmids are the circles of DNA linked to antibiotic resistance. Well, prokaryotic cells do have plasmids, whereas eukaryotic cells do not. And that's an easy way to identify what type of cell we're looking at in an exam.